And good evening, everybody. Thank you for joining us tonight. My name is Antoinette Handley. I have the honor and privilege of serving as the Vice Dean of Graduate Education here in the Faculty of Arts and Science. And I'd like to begin by acknowledging the land on which the University of Toronto operates. As many of you will know, for thousands of years, this land has been the traditional home of the Huron-Wendat, the Seneca, the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from all across Turtle Island. And when we make this acknowledgement, we express our gratitude for having the opportunity to work on this land. It is my very distinct pleasure to welcome you this evening to the 2026 Martin Lecture in Ecology and Evolutionary Biology. The Martin Lecture Series was created in honor of the Martin family and their exceptional generosity to the Faculty of Arts and Science. There are many of the Martin family with us here this evening, and for generations, we have been extremely fortunate to maintain a strong connection to that family. Martin and Ron Martin's 4 children graduated from the University of Toronto, as did all 6 of their grandchildren, and we're delighted to see some of them in the audience with us tonight. But this family connection stretches back as far as the late 1800s. All told, some 60 members of the extended family have attended U of T. I'm not sure that's a record any other family can match. So the Martin and Ronald Martin Graduate Scholarships were established through a generous bequest from Mary and Ron Martin, who were champions of higher education. For that, we are extremely grateful. These scholarships help us attract the most promising students from across Canada and all over the world. The scholarships are awarded to top graduate students in 4 disciplines: astronomy and astrophysics, ecology and evolutionary biology, physics, and public policy, which just happen to be the professional specialties of Mary and Ron's children. So the event we're here for today, the annual Martin Lecture, rotates in focus amongst those same 4 departments. And every year we strive to bring you compelling talks that explore the most relevant and fascinating issues of our day. I can assure you that tonight will be no exception. I am so excited that we're here for a discussion that will address some of the most urgent problems in medicine, namely antibiotic resistance. And we're very lucky to have Professor Paul Turner, an evolutionary biologist and virologist from Yale University, to speak with us this evening. His research lab, as I'm sure Joel will tell us in a moment, focuses on virus ecology and evolution, including how viruses emerge, how they move to new host species, and how they adapt to environmental challenges. Professor Turner's research group is studying how to use viruses to treat antibiotic-resistant bacterial infections. Now, as a political scientist, I'm already in dangerous territory. So without further ado, I will turn things over to Professor Joel Levine, Chair of the Department of Ecology and Evolutionary Biology.
Thank you, Antoinette. Antoinette and I were talking just before the evening began, wondering whether all of the Martins who are here are in the first 2 rows. Is it fair to ask whether the Martins would hold up your hands? Yeah, see, there's more. Thank you. This is what we were guessing. And we want to say thank you. Thank you for this lecture series. So good evening and welcome to the 2026 Martin Lecture in Ecology and Evolutionary Biology. It is a pleasure to introduce tonight's speaker, Dr. Paul Turner, Rachel Carson Professor of Ecology and Evolutionary Biology at Yale University, Professor of Microbiology at the Yale School of Medicine, and Director of Yale Center for Phage Biology and Therapy. Professor Turner's work has reshaped how we understand viruses. They are not simply agents of disease. They are elements of evolving systems that can be studied, anticipated, and in important ways controlled or guided. Professor Turner trained at Michigan State University under Richard Lenski, a pioneer of experimental evolution. He He has built a career at the intersection of evolutionary theory and microbiology. His research focuses on viruses and their interactions with hosts, especially bacteriophages, the viruses that infect bacteria. He examines what these systems reveal about the evolution of infectious disease and antibiotic resistance. What distinguishes Professor Turner's work is a conceptual shift with practical consequences. In the clinical setting, evolution shows up as a problem in the treatment of infectious disease. For example, a bacterial pathogen undermines drug therapy by evolving resistance to the drug. But Professor Turner is at the forefront of evolutionary biologists who have asked if the principles of evolution could become part of the therapeutic solution for treatment of disease. A key moment in this shift came from his work with Benjamin Chan and colleagues on bacteria called Pseudomonas aeruginosa. They demonstrated that a bacteriophage could be selected to target a bacterial mechanism of antibiotic resistance. The bacteria were using a molecular pump to expel drugs, the antibiotics, but when the bacteria evolved resistance to the phage, they did so by altering the pump, rendering the infectious bugs more vulnerable to antibiotics. The bacteriophage on the one hand and the antibiotic on the other placed the infectious bacterial pathogen in an evolutionary checkmate. This study provided a clear and elegant demonstration of a powerful idea that antibiotic resistance is not simply a barrier but a trade-off, and that such trade-offs can be exploited. Rather than attempting to eliminate resistance to drugs, Professor Turner's work has shown that we can design therapies so that every adaptive move given by a pathogen carries a cost. Building on this insight, Professor Turner's work has helped transform phage therapy from a largely historical or experimental approach into a contemporary, conceptually grounded strategy. His lab has shown how evolutionary thinking can be used to design successful treatment protocols that not only reduce bacterial populations, but steer them toward states that are less virulent and more treatable. His contributions have been widely recognized. He is a fellow of the National Academy of Sciences, the American Academy of Arts and Sciences, and the American Philosophical Society. He has held numerous leadership roles at Yale and in the broader scientific community, and has contributed to national and international efforts to understand and respond to infectious disease. This perspective resonates strongly within the Canadian scientific tradition. Research linking evolutionary approaches to public health issues may be found at universities across the country. After tonight's lecture, Professor Turner will be joined by members of our department, EEB, the Dalla Lana School of Public Health, and the University of Toronto School of Medicine, who are all a part of this Canadian research community. Professor Turner's work sits squarely within and extends this tradition. It brings together deep theoretical insight and practical application in a way that speaks directly to challenges that science in Canada and the rest of the world have long engaged. But approaches like phage therapy also raise important questions beyond laboratory and clinical protocols. Using viruses as therapeutic agents can challenge public intuition and may encounter another form of resistance, particularly in communities where trust in medical systems has been shaped by historical inequities. As these therapies advance, their success will depend not only on scientific rigor, but on thoughtful communication, community engagement, and a broader public understanding of evolutionary principles as a tool in medicine. This brings us to the theme of tonight's lecture: leveraging evolutionary trade-offs in the development of phage therapy. Both in the lab and in the clinic, Professor Turner has shown us that if we understand evolution well enough, we don't have to fight consequences such as antibiotic resistance blindly. Instead, we can design conditions that define a path of least resistance such that even deadly pathogens are steered into losing strategies. Please join me in welcoming Dr. Turner.
Okay, so it is a distinct honor to be here. I'm very delighted over this invite, and thank you to the Martin family for your generosity. All right, so that was a great introduction. That gave away some punchlines, but that's okay, because of what I've learned in the classroom is reinforcement through hearing things twice, right? No, I'm just kidding. So now, you've heard the title of the talk. These images, what to make of these? So they really are a way for me to remind you of a classic way that phage therapy has been done since the early 1900s. In a lot of ways, we still rely on this. We look around the world in natural environments for phages, which are incredibly abundant. So you'll hear more about phages in a moment. But you can find them in natural environments. You can find them in the human home. You can find them in this auditorium. And when we isolate them, they just might have some properties that are useful for biotechnology, including maybe developing new human therapies. So that's a picture of one of the phages that we literally found in a lake in Connecticut. This is Ben Chan isolating from a much more interesting location than Connecticut, that's Mexico. But a lot of the phages I'll talk about today really have this life cycle where the phage is infecting a bacterium, taking over that bacterium, turning it into a production factory for virus progeny that accumulate, and then the cell dies in the process. So this is the beauty of this kind of a drug, is that you're targeting a bacterium in this case, and your drug is making copies of itself. Ultimately, what we're finding, and what people have found through phage therapy research through the years, is you have an immediate possibility of launching into treatment. And I'll remind you, even under the simplest of equipment and insight, in the very early days of phage therapy, this was happening. I think now we're at a moment we have to bring more rigor and understanding to something that has shown a lot of promise, but we want to see just how far we can push it. So the outline for tonight is that I will talk more about phage therapy. I'll talk about, to me, my opinion of a very big barrier to phage therapy is what you heard in the introduction, is that the bacteria can evolve resistance to antibiotics. Why would we not think that they can evolve resistance to phages? Phage steering is the way that we're popularizing that if the evolution happens, it's going to go down a path that I prefer and I'll design, and we see that in the lab. And it's interesting that we see the evidence of it in the clinic. But there's a big remaining number of things to do next. And so I want to make sure I capture enough time at the end to impress upon you that there's a lot more that we need to know and want to know about this to move forward. So bacteriophages are indeed viruses of bacteria. They were missed for a long period of time, so microbiology is not, it's not a very mature science, right? We only had the tools, microscopy, to see things. But this is what we missed a lot, was the virus world, because you really need special equipment to see things that are this small. And yet the elegance, I would say, of the forms of viruses that you find in nature, including bacteriophages, is quite impressive given how small they are. We have these awesome tools here that you can use, all sorts of amazing cryo-EM electron microscopy tools to reveal structural details of phages. So as an evolutionary biologist, I care about patterns and relatedness. What you're seeing in this map is there's a common ancestor that gives rise to all these bacterial types that we have known and found in nature, and they're color-coded by their different family groups. And on the edges here, we're seeing a handful of these phage types, and one thing that you'll immediately see is that there's not a one-to-one correspondence historically in the evolution of life and what can parasitize it. In this case, phages can exploit very specific species, but they can also have close relatives of phages that are exploiting bacteria that are very, very distantly related to the species that they're infecting. So a lot of this is very hard to figure out patterns-wise on Earth what's going on here. So just keep in mind that there is an amazing diversity of bacteria, an abundance of bacteria on Earth. We are learning more and more as we discover more phages and filling in the blanks of the families of phages that we discover, which are often new ones. And it's not clear how they're exploiting closely related species versus distantly related ones. But that's all I'll say about that topic. I will now move on to really the core of what I'm presenting, and that is, can viruses solve problems? And back in the day, one of the co-discoverers of phages, Felix Durrell, did his work, and some of that work was done in Canada. He lived part of his life in Canada. He was a faculty member at only one institution. It happened to be my institution. He didn't get on with his chair of his department, and I think he left the university after a short period of time. We won't get into that. But he recognized something very early on when he first saw phages in his research, is that they should have potential to control infections. And he did this work, my mic, I'm not sure if my mic is, okay. He did this work in experiments purposely infecting animals and people, and then chasing those infections with phages to see if phages could control the infection, which you can't do now, just in case those of you who were befuddled by that. No, you can't do that research anymore. But he learned a lot, and he showed a lot of promise very early on. So the point is, soon after phages were discovered, they were immediately used in a phage therapy context to see how useful they could be. So now we fast forward to the current day. You haven't heard that much about phage therapy in many parts of the world, the Western world especially, whereas in lots of places, they never really adopted antibiotics as a go-to, either because it was too expensive or for other reasons of access, and that still exists today. So antibiotic resistance and its spread is a big issue. There are places where still people don't really have good access to antibiotics. Nevertheless, the overabundance, one can say, of the use of antibiotics has created this problem of increasing evolution of antibiotic resistance in bacteria, and a projection map like this, which is talking about decades not that far from now, where the goal of this is to impress upon you that human mortality will be increasingly affected by antimicrobial resistance as we move forward. Unless we do something to help remedy this problem, that I'm sorry that the youngest members of our audience, they may live in a world where antibiotics just aren't working as well as they do today. And the projection map looks very uneven for its mortality, but that's just because of the projected population sizes of people living on these continents are different. This is a global problem. And the enduring question now is, okay, if we have this problem and we need something to either replace antibiotics or help them work better, can phages do this? So let's start with a very important early problem that Durrell and others of phage therapy research saw, is that most lytic phages are very specific in what they kill, was the idea. And this creates a specificity issue that if you take these three phages and imagine all of you have the same bacterial illness, but you have different strains causing it. And so if I was going to try and use phage therapy to help you, I may not be able to treat all of you with the same phage. And therefore, immediately, it became popular to mix phages together as cocktails in order to sort of cover more of the strain space. And yet you'll see you can still miss things. Strain 4 and strain 7, for example, here are not being covered by this 3-phage cocktail. So this was an immediate liability as phage therapy started to move forward. And it continues today, this idea that specificity is a big deal. And I'm here to tell you, I don't think it's as big of a deal in many of the bacterial targets we want to tackle. So here's some evidence from it from a startup company that we had that's no longer operating, but Felix Biotechnology and some of our research involved vast phage libraries here along this edge, taken straight from natural sources, water and soil. And it so happened that these phages were able to kill an important multi-drug resistant, increasingly multi-drug resistant bacterium called Pseudomonas aeruginosa, which the World Health Organization has told us we need to be very worried about it among the 6 major types of bacteria that will cause this increasing rise in antibiotic resistance. So if you take P. aeruginosa strains from infected people in a clinical setting and you challenge these phages to kill them, and you recognize that everywhere on the left that you have a phage and you have a dark blue square, it's killing the bacterium at the top of the column. And there's a lot of blue. So you can find at least phages in this system that cover a pretty appreciable genotype space in their target bacteria. I'm not claiming that that's the case in all systems, but this is not like the early too specific to be useful data that Durrell and others saw. So here's another thing that gives me some hope in terms of the target specificity limit problem, is that increasingly you can have better tools in synthetic biology to help you design the phage you want. And we were happy to collaborate with New England Biolabs on a paper that was published recently, where this was entirely the goal of New England Biolabs to sell you a kit that you use in your laboratory. So what they would rather you do is take some phage of interest and the genes that you want to assemble into a phage, and you put it on a series of plasmids, but you do a Golden Gate assembly process, that's not really shown here, in a test tube with a surrogate bacterium that these phages can't normally infect. And that would be E. coli, and it would pump out the particles that you want. So we showed this in a paper recently, and the point is that it works very efficiently. And again, you're designing phages using a surrogate and then using them in a system that is different. So that gives me hope that if you want to make phages broader, you understand enough about the underlying mechanisms and you build it in to the process. So that's the good news of conquering an old perceived problem. And the bad news is that the evolution of phage resistance is now convinced you a very big deal, that if you are using phages for any biotechnology, really, I'm talking most about therapy here tonight of humans, but people are trying to design phages to break up sludge in pipes underneath streets, like in Toronto, because that can be a big deal for moving things through pipes. So there's lots of ways that you can use phage biotechnology, but the problem is if you deploy it and the bacteria are not going to be happy about it because they're dying. So they undergo strong selection pressure to change evolutionarily to overcome that problem. So again, here I'm showing the very familiar by now diagram of phage replication. Now on the right, this may look a little mysterious to you, but it's a piece of equipment that we use in the lab all the time in microbiology now. We're using light scattering, or OD600, optical density, as a surrogate sort of for, or a corollary for bacterial growth. So you essentially can track automatically in these automated spectrophotometers the growth of a bacterial population, and you see its characteristic increase, and it runs out of nutrient in the little well that you're monitoring. But you can do that 3 times, you get the same result. So if you had a phage in that well with them, eventually the phage would overtake the bacteria numerically and kill them, and you should see this initial growth of bacteria, and then they crash. They sort of go locally extinct. Except by the next morning, you come in and you look at your data and you start to see this. So what is that? Those are mutants that were resistant to the phage, and they were there from the very beginning. But they were very rare, and it took enough time for them to be benefited by the fact that their brethren were dying, and then by the time you come back, you see an outgrowth of them. And it happens again and again and again. And it could be different points in this replication cycle, where I'm drawing an X, at which those mutations are allowing the bacteria to overcome the phage. So that's a variety of ways it can occur. So ideally, as an evolutionary biologist, and many of you in the audience, you appreciate host-parasite theory, and what I'm going to show next is that, well, perhaps if this is happening, you can rely on phages to overcome the problem. And essentially, they will evolve or co-evolve with their bacteria to become better at killing them. And this takes us to the very enduring kind of metaphor here, negative frequency-dependent selection. So what does this mean? Parasites and hosts can get locked into a battle called an arms race, where there could be better defenses on the part of the host and better ways to attack or infect on the part of a parasite. And so they kind of fluctuate through time numerically, but they're kind of chasing one another, and they can't break apart from this relationship. And this has been often called the Red Queen effect or the Red Queen hypothesis. This was really a paper about avoidance of extinction, but it's been used to talk a lot about how host resistance and parasite virulence can be in a lockstep. You'd expect phages to be able to co-evolve with their hosts. And you can see this in the laboratory. So some of this work by Buckling and Rainey from some time ago, I don't think they did any sequencing because sequencing wasn't as common back then, but they grew bacteria and they grew phage together, but I'm showing you dynamically here some traits of the bacteria. Increasingly, they are resistant to the phages that are co-evolving alongside them, but the phages are increasingly able to infect those resistant bacteria. So that shows evidence for an arms race. So they're basically locked into a battle that they're not causing each other to go extinct, but they're having to keep pace with one another evolutionarily. So that was back in the early 2000s, and now we have entire laboratories that are devoted to discovering new mechanisms of phage defense and other ways that bacteria defend against what are called mobile genetic elements. And this is a pretty crazy, complicated diagram. I'm not going to walk through it. It's just to impress upon you that there's lots of information coming about. I mean, literally week by week, it's fantastic to me that things that have evolved in bacteria for defenses we've missed because we didn't know how to look for them properly. And there is evidence, I guess I'll add this in, it's not really on the slide, but some of your innate immunity genes seem to be hallmarks of what bacteria evolved long ago. And they never disappeared. Eukaryotes, like you, inherited them. So one can then ask, well, how the heck are phages making a living on this planet? If there's so much that is against them, and how could we be so ambitious to use phages in biotechnology given what's stacked against them this way? So how would you address the problem? How could you limit evolution of resistance if you have bacteria you want to kill? You could develop therapies that minimize the evolutionary potential of the bacteria somehow. And cocktails might do that, so I'll talk about them a little bit later. but we don't study that as much. I'll focus mostly on the lower bullet point. I'm just going to toss my hands up and say the bacteria are going to evolve. I believe that they can do it. I know that they're going to do it. So I want them to do it on my terms. And this is what you heard in the introduction and what's shown here. Ben Chan is the one who has mostly studied this with me for the last 13 years now. He's been a member of my group as a senior scientist. So it's a busy diagram, but I'll walk you through it. The yellow triangle, which some folks in the audience have undoubtedly seen, is that if you have bacteria, and if you know enough about them that they have a mechanism such as an efflux pump that is pushing antibiotics out and making them multi-drug resistant, because this can work against different drug classes. And this is a hallmark of some of the bacteria we're most worried about. But if you have a phage that is somehow specific to binding to efflux pump proteins, and if it's a lytic phage that's going to kill the bacteria, this creates quite a selection pressure for the bacteria to solve this problem. And there may be plenty of mutants that just pop up in bacterial populations that have altered efflux pump proteins that are maybe less functional, or they just don't have efflux pumps at all because they're born without them. So they should be the ones that are enriched when you deploy a phage, and in this way, and selection pressure will push them down into this corner where they might have solved the phage problem, but they become resensitized to antibiotics in the process. So that's pretty nice if we can get that to work in the clinic. And the purple triangle would be toggling virulence, that if you have a worrisome bacterium that has a capsule which helps shield it from your immune system cells to detect it, if you have a phage specific for that, it'll exert selection pressure for the bacteria to lose that capsule, and your immune system cells will better clear those bacteria. So this is conceptually a way of thinking, how can you deploy phages and push them to steer the bacteria in the way you want for evolution? So I don't have time to go into some specific examples of virulence toggling. You can take a look at this paper that was led by a senior scientist in my group, Katie Courtwright. Shigella is a pretty nasty diarrheagenic bacterium, and it moves from cell to cell in your intestines because of some proteins on its exterior. Well, we found in Mexico with colleagues there, phages that naturally exploit these binding sites. So the Shigella bacteria solve the phage problem by losing those proteins. And this makes them lousy at moving between cell to cell in your intestines. So this could be one way to toggle virulence. What I'll talk about a bit more is phage-antibiotic synergy, where what I explained to you is essentially a way that you would think antibiotics and phages could work better together than alone. And that has been seen a lot in the literature without knowing mechanistically why. One example would be the mouse experiments done by Smith and Huggins, where they got huge signals for this in the 1980s, but they didn't quite know what was happening. So I'm trying to explain to you that this is not something that I created. This is something that has been observed, but to popularize it and try to push it into the clinic and to do this phage therapy in a more kind of evolution thinking enlightened way is what we've been trying to do. So now I'll talk in some greater detail about work that was continuing with a former postdoc, Alita Burmeister, but some of the work that she did with me and my group was looking at the Toll-C efflux pump. This is a beautiful structure for which we did not create it, but you should be impressed with it. But it functions the same way as the efflux pumps that I showed you in the cartoon. And the point is, we have a phage that is very good at interacting with the outermost proteins of Toll-C efflux pumps to push or steer bacteria down the right path. So let's talk a little bit more about this, of phage steering with this example. So our goal is, if we can identify phages that drive antibiotic resistance trade-offs, we would like to develop these and use them in the clinic. So an example is this phage called U136B, and it selects for antibiotic resensitivity in E. coli. I already told you it binds to efflux pumps. So let's look at some data that if you consider its ability to drive tetracycline sensitivity, what do I mean by that and how would you see it? In the absence of tetracycline, in the absence of this antibiotic, there's a way of doing a dilution series with bacteria to understand them numerically, and you could look at wild-type bacteria and an engineered Toll-C knockout that you wouldn't expect it to grow well in the presence of an antibiotic, and a spontaneous phage-resistant mutant of these bacteria resistant to this phage. They grow equally in the absence of antibiotic, but in the presence of tetracycline, you'd expect that Toll-C mutant to do very poorly. And phenotypically, or the same traits are seen in this spontaneous phage-resistant bacterial mutant. So that's good news. It says to us, oh, okay, it is using the efflux pump, And the bacteria can gain resistance to the phage, but they give up resistance to tetracycline and become sensitized. But as I like to say, in this system, the plot thickens, because a lot of viruses bind to more than one thing on the cell surface, including this phage. So it happens to also use as a receptor something that fortunately drives a different kind of trade-off. So there's this substance around the exterior bacterial lipopolysaccharide, or LPS, that is extremely abundant, structurally helpful for the bacteria. And it can be that phages come in and they bind to some midpoint of the LPS. So imagine that this is the surface of the cell. It's almost like this is growing up like a tree from the cell surface, but the whole cell is covered in this. And what we found is that if mutants arise in these bacterial populations that lop off the top, overstory? What is it? Must be some plant biologists who can help me with this. So they lop off the tops of the trees, and this is a way that they gain resistance to the phage, but it causes them to be more sensitized to a different antibiotic called colistin. So this is one phage that drives two separate trade-offs, depending on what the phage binds to and what the bacteria will do to solve that problem genetically. So then we did a kind of a classic approach, identify rescue mutations that would be responsible for this phage resistance. We wanted to know how often does it happen in Tol-C versus how often does it happen in LPS. So you just grow bacteria and you grow them up, and then you can plate for resistant mutants, and you might see a lot of them on a phage plate because it happened early in the growth of the culture, or maybe it happened very late and you see very few of them, but you can isolate them and take them on for further research. So this is a busy slide that shows that. Starting on the right, in this paper in 2020, we isolated 20 separate bacterial mutants that were all resistant to this phage, and we did all the whole genome sequencing to tell us where in the genome is this responsible. And this behaved perfectly in terms of everything in purple are mutants that have differing Tol-C mutations. Everything in green are those with LPS mutations. And the change in the minimum inhibitory concentration of tetracycline or colistin also worked very well in terms of the consequence on average of those mutations. But here on the left is something that we wanted to do further. We wanted to examine, well, look, if they could change Tol-C or they could change LPS, would it matter for their general growth? So why would we care about that? Mutants arise in the presence of other mutants. And whatever mutants are least costly, those are the ones you'd expect to win. So one can use this automated spectrophotometer, and we get our growth of our wild-type bacteria in black. And all these Tol-C mutants seem to be growing just as well in the absence of any perturbation, no phage, no antibiotic. They're growing fine. But again, under normal conditions, these LPS mutants are not very happy campers. They are debilitated in their growth. They might have gained phage resistance, but they're pretty poor at undergoing normal growth. So what does that mean? If you were going to go to Las Vegas, or the Canadian equivalent, which I'm sorry I don't know, is which of these mutations would be most likely to occur in a treated patient? And I would bet on the ones in purple, because they are competitively superior to the other mutants that I'm telling you are very abundant in the system. So the way to test that is you do experimental evolution. You take the bacteria and you evolve them in either the presence or the absence of the phage. So let's take here in red, if you just evolve them for 10 days in the absence of the phage, and you look at something that you didn't select for, and that is resistance to tetracycline, they don't really change beyond wild-type level. They might fluctuate a bit, but they kind of maintain wild-type. But if you grow them in the presence and evolve them in the presence of the phage, they start to move downward in this trait. They start to lose that trait. And that's because I'm not showing you the accompanying genetic data to say that they were fixing Tol-C mutations. They were not fixing LPS mutations to solve this problem. Plenty of LPS mutations probably occurred, but they were outcompeted. So now we know, at least in this system, we can predict which mutations are going to be the most common ones, and we might gain some confidence about what we would see in the clinic from that. All right, so now I'm going to go through a series of some new data, and I'm beginning with this very complicated cartoon, so you can take it in as I take a drink of water. Emma Kane is one of our post-grad associates. So she finished her bachelor's degree, and she's going on to med school soon. But she's working with us for a year. And what I'm showing you here is a cartoon of a plate with 96 wells, or approximately that. And basically what you can do is, along this plate, you can seed the whole thing with bacteria, but you can look at different levels of antibiotic and different levels of phage as you move across or down the plate. So there's going to be some combinations in the middle that I'm highlighting in this rectangle where you just might see something interesting. You might see that in the combination of the phage and the antibiotic, they kill the bacteria more than you would expect based on either of those alone. And you'd get a big signal for it if I'm right in discerning here. So this would be if bacteria are growing in this level of phage, and they're growing fine in this level of antibiotic, but here in the middle, they are crushed in the presence of the two. Okay, so this is what we would look for from the expectation that if I deploy a phage, and I just want it to work straight out of the box better with an antibiotic, I want to do assays like this to tell me, is it possible? Just so you're keeping track, I gave you an example earlier. If the phage is pushing the bacteria to become resistant to the phage and then convert to being sensitized to an antibiotic, here's a little bit more in the realm of, is there just something about antibiotics and phages working together that beats them working alone? Okay? So Emma did this, and she found something that surprised us. So first of all, she took a statistical analysis to it where others don't tend to. It's called a Bliss index. But essentially, she can do this for ciprofloxacin and aztreonam and 3 other antibiotics. And essentially, it's giving a score to this possible synergy versus antagonism versus neutrality that's additive. And she starts to see, whoa, there is a lot of evidence for this one phage that she was exploring that is able to kill P. aeruginosa, but when you put it in the presence of some of these antibiotics, 4 out of the 5 show her synergy. So that would be good news for this phage if you wanted to deploy it alongside antibiotics, and there would be a variety of them. Turns out that that phage is something a little complex, and it's newly discovered in phage biology. It's called a jumbo phage. So she measured this for a phage that does something really cool when it comes into a bacterial cell. It's a cartoon of it here on the right. It comes in, and it forms a compartment, unfortunately called a nucleus because it's a prokaryote, but I won't say anything further on that. It is a little confusing, but just think of it. It's a compartment that is walled off from the knowledge of the bacterial cell, understanding that phage replication is occurring. So it's kind of automatically defended against a lot of anti-phage defenses in the cell. So that has something that the phage has as its disposal, but I actually don't have a great reason to think that that particular kind of phage should synergize with antibiotics in any way. I'm not sure why that would be. So we're looking around for evidence and finding that a reminder is under sublethal antibiotics, bacteria do weird things. Sometimes they'll elongate without dividing because the nature of the antibiotic would be to stop them from growing rather than killing them entirely. And it turns out, maybe, and so this is where stretching plausibility, because I've never studied it, is can you create a better target for phages to even attack cells if they're elongating? That's a simple physical process that people may have missed often when they see phage-antibiotic synergy historically, is that bacteria do strange things. If the antibiotic is not killing them outright, they might do things that would make them more vulnerable for phage infection, okay? So we have to think about that moving forward. And the point of all this is to say, pick the right phage, and it could matter for your success of what you want to achieve. So along those lines, what is any evidence that any of this would work if we tried doing it in the clinic? So that's what I'm going to shift to next. If you're keeping track, you'll notice that there's something about phages that I'll say is true for most viruses, is that they tend to use binding sites of things that are very important to their cells, and therefore they're reliably there. And we call that constraint in evolutionary biology. If you need something that you tend to see it evolve and it's maintained in lineages and in species and so on, then that's a great target for a phage or any other virus, if it's attacking a eukaryote or whatever, because you tend to find it on the cell surface. So there's something that bacteria do that a lot of organisms do, is that they need to move around their environment. So phages often exploit conserved binding sites or receptors on bacteria, and bacteria use motility mechanisms to move around their environment and access resources and to enter new environments. This is a very elegant structure, colorized of a type 4 pilus. So why would you care about type 4 pili? These are extended and retracted from bacteria, and they use it to do twitching motility or to move across surfaces. So when I first started studying phage biology in a postdoc, I saw this immediately in a phage bacteria system we were studying because the bacteria happen to be plant pathogens. And these bacteria ordinarily are covered with these pili because when they land on a leaf surface, they're trying to twitch across the leaf surface to enter the plant through the stomata, okay, the holes in the leaves. So Pseudomonas syringae is an incredibly abundant pathogen of plants. You find it readily, and this is what it does to enter plants. So what we saw in the laboratory, because we were not growing any plants, is that we would challenge our bacteria to overcome phage attack, such as phage Phi6. We were studying that, and this phage binds to type 4 pili. So the way that the bacteria evolve an escape route is to be a phage-resistant mutant that doesn't make pili. Okay, makes good sense. Hard to get away with that in nature and be a plant pathogen at the same time. Okay, so we have not studied that under natural conditions, but in a way, what we're showing is that if that occurred, it breaks the Red Queen, basically. So there's not going to be any more arms race because there's not necessarily something that the phage could bind to, but the bacteria pay a very big consequence. They have to occupy a different niche space. They can't go on being a plant pathogen. So this is actually the seed of where I started thinking about phage steering. And for the young scientists in the audience, always hang on to your ideas because you never know, decades later they might come into some utility. Why we care about this in the clinic is that, again, P. aeruginosa has a lot of pili surrounding the cell, and it helps it adhere to surfaces, including your cells, causing chronic infections. It also can be associated with certain virulence factors being secreted, like piocyanin, which is a very destructive thing for your cells. And if you have a lung infection that is chronic with these bacteria, it can destroy enough of your lung tissue that you would need a lung transplant. Okay? So for people who are vulnerable to these types of infections, this is a very big deal. In principle, we could use a phage like this one called TIVPH6 or type 4 pilus H6. Its picture is here. A relative of it is here from another group's work. Their pictures are prettier, so I show it. And the idea is, all right, if you deployed this and you wanted to target bacteria that have type 4 pili, could you use it? So I wouldn't be standing here had it not worked. This is a pretty crazy, colorful diagram, but all it's really showing, again, is a structure of the type 4 pilus color-coded so that it's impressing upon you lots of underlying genes are responsible for making type 4 pili. And any of them could, in principle, be broken that you don't construct pili, allowing you to be a phage-resistant mutant. So we would expect a whole bunch of different mutants to evolve spontaneously in the laboratory when we challenge them to be resistant to the phage. And something they all have in common is you measure their twitching motility or their ability to move from the center of a plate outwards, and they're all bad at it. So that's like the phenotypic proof that they have less functional pili, and yet they're gaining phage resistance, but they're giving up their ability to move. And in this case, we would care about their ability to attach to cells and cause biofilms as something they give up as well. So now we go to clinical data, and the red arrows are showing in a study we published, when was that, last year, in a cohort of 9 individuals who were all born vulnerable to chronic infections of the lung. These people all have cystic fibrosis. And they came to our attention as emergency cases because they were all slated for either lung transplant or things were looking even worse than that, and they had run out of options because their bacterial infections were multi-drug or pan-drug resistant. So I'm showing in arrows all the times that we deployed either this phage alone or as a cocktail to help these people. So here on the right is the good news first, is that if you track in these individuals before the therapy, and then 14 days after the therapy and 30 days after, they all experienced inhaled therapy. So they're inhaling droplets where phages are in the solution. And those travel to their lungs, and the hope is that it would kill the chronic infection of bacteria. So we can unpack that later if you want as a hope. But essentially, you can measure how much of these target bacteria are still in their lungs by looking at their density from sputum or mucus samples that they cough up and provide as samples. And on the right is the good news that in all 9 of these individuals, post-therapy, they trended upwards in their lung function, which is abbreviated here as FEV1. It's the ability to force air out of your lungs when you're told to do so. Now, you have good lung function compared to the vast majority of these people. The whole reason we were helping many of them is that they were already slated for a lung transplant, but you can't ethically do the surgery if you have an infected lung, for fear that you remove, that could be the goal. If you just remove one lobe, you could still have that infection jump to the other lobe. So no surgeon is allowed to do that, and people like us can be called on as, okay, can you at least reduce or control or eliminate that infection so we can go forward with a lung transplant? So you may not be impressed by this increase. That's because these individuals had lousy lung function and they were never going to get it back. But now they can undergo a lung transplant. So another way to summarize these data, well, first I'm going to extend out this table. Before therapy, we would take clones from each of the individuals, we would grow them up in the lab, and the open circles indicate that these 3 clones coming from this person are all sensitive to this phage. Otherwise, we never would use it in therapy. After therapy, post-phage therapy, the filled circles show that now we're pulling out more and more phage-resistant mutants, which I will claim cannot be an accident. It's because we put the phages in that you would expect this to occur. And on the right, it's a little hard to understand, but I want to make sure before we treated these individuals, you saw very little pre-existing resistance to a phage. And now that's in abundance afterwards, and the overall number of infecting bacteria per person has dropped tremendously into a range that, at least in principle, the human immune system can start to work with you to push that infection out. Because if it's too many bacteria, it's too raging, that'll never occur. All right, so the further good news for this cohort of 9 is that when we go and measure things that should be associated with this phage steering the bacteria down a path that we prefer, I would think that if I take bacteria pre-therapy from patient 2, and I measure the ability of those clones of bacteria to attach to cells of human origin in the laboratory, they may have a range of abilities to initiate biofilms, but plenty of them are very capable of doing it. And after therapy, it all troughs out. So that's what I would expect if their type 4 pili are missing or non-functional. The decreased piocyanin production could follow. In these 3 patients, we saw that because these pili can be important for secreting the virulence factor. And if you take the post-therapy bacteria, you grow them in the lab, spin them down and discard the cells, you can take the supernatant and put it on human cells that are mimicking the airway, the epithelial airway, and you should see that those cells are less they should show fewer inflammatory markers because there's less piocyanin being produced by these bacteria, and indeed, you look at markers like IL-8 and IL-6, and it goes in the right direction. The last thing we would want to know is if you're deploying this phage and you're getting this result, that it's really due to the type 4 pili changing. And the only way to do that is to do whole genome sequencing and map what's going on. So now, these pictures are much prettier, and they come from Jean-Paul Pernet's paper describing the first 100 cases that they did in Belgium. But they basically deployed often type 4 pili targeting phages as well, and they would map for patient 54 here and patient 30 who received a type 4 pili targeting phage where in the genome did the changes occur, and they're often seeing that the changes that give the phage resistance are these genes that are responsible for type 4 pili production. So things start to match up for genotype-phenotype mapping. All right, to end, I'm going to talk about what's next, and I'm going to go rather quickly because I know we're running out of time, and there's a lot of what next, so we can talk about it during the panel if you'd like. One thing is, we are not so foolhardy to think that we're actually attacking the entire bacterial population in a chronic infection. We're often going with the information very dedicatedly given to us by a clinical microbiology lab telling us what is the major infecting strain. And they don't tell us much about the other close relatives of those infecting bacteria that might be in there as well. And it is well known that when P. aeruginosa colonizes the lungs of a vulnerable person, like a CF individual, it will progress to a chronic infection, and I'm showing now very different colors and structures because that's what happens. The bacteria diversify. And yet, if we are called upon to help in a compassionate therapy case, it would be a clinical microbiology partner telling us, well, what is the strain we need to worry about to match to our phage? And that's what we go forward with, basically ignoring all the rest of the variation. So does this matter? There's one way to measure it very easily, and I have to introduce the measurement. You can grow phages on some bacterium that's a model, and it has very robust growth, and then you can compare its growth on some other strain of bacteria of interest, and that's called efficiency of plaquing. So you sort of do a dilution series, and you're kind of looking at how well do the phages form these plaques or the killing zones of bacteria, and you can relate your phage ability to grow on a target strain compared to something that's a model. When they grow equally, in the next slide, I think it's going to be a 0. Okay, so there's no difference between them. So one of my current graduate students, Noah, who happens to be Canadian, but he did an amazing study here with a set of strains that came from Georgia Tech and Emory University called the Diggle Collection. Steve Diggle isolated these. These are individuals who are still alive. They have cystic fibrosis, but they've given up strains that had colonized their lungs for future research. And we looked at some of these, or Noah did, and he found that individual 1, 2, 3, and 4, if you look at 4 phages that are all binding to the same thing, lipopolysaccharide, some portion of it, you find a very big difference for the pre-existing variation that you would expect to see in an individual like this, for the ability of some phage to kill one variant versus all the variants that are there. So here's the best news, right? For whatever reason, this individual, all 4 of these phages, they grow equally as well on these P. aeruginosa strains as they do on some model strain. That's the good news. The rest of it's bad news. I mean, either they're not doing well at all, or this is not what you want to see. This is like all over the place. And to emphasize, it's like, well, maybe we would target this one, or the clinical microbiology lab would tell us to target this one, or this one. And so there's all this other variation that we expect to not be successful, and yet, remarkably, our research goes forward and it works. So you can ponder that. This is a little challenging to say, but it's a little bit more in this. If you have some clones that I just showed you of bacteria that are phage resistant, can you hope that at least they're antibiotic sensitized? And you could look for a negative correlation. So only in this population of bacteria coming from CF individual 1 are all these red things lighting up in the upper left corner. For all the rest of them, it's quite a mishmash. And we cannot be thinking that we can universally cover all the variation when you deploy this kind of technology. All right, to end up, there's different ways to deliver phages. You can do it directly to the site. So we've treated aortic arch replacement problems where a chronic infection forms on an artificial indwelling device, which would also be a prosthetic joint. And you can directly go to the problem with a syringe. It's very straightforward. I showed you already pulmonary infections. You can do inhalation therapy. And others have done that. In principle, you could do bloodstream delivery for almost anything, because you would want to do that if you had sepsis, which is a bloodstream infection. But there's plenty of evidence that if you just stick a needle in somebody's arm with a phage, it'll travel to the side of the body that it needs to, and it'll kill the bacteria. Okay? So the problem, I would say, is this. If you do that, would you or would you not, depending on the phage, mount a bigger immune response in the patient to attack the phage? Arguably, I'm not a physician, many of my friends are, and they tell me maybe we need to think about bloodstream delivery because of this inherent problem. So hold on to that thought. The main point of showing you this slide is to say that we need to do more research and understand more of the human immunobiology response during the therapy. So we work with modelers like Joshua Weitz, who does phage antibiotic combination therapy models, where also he's building in the immune system and how it could be working as a trio together with a phage and an antibiotic to cover a problem. Some of the work we've done with Jen Bomberger at Dartmouth was quite fascinating to me, but this is only one of several papers that have been published now, showing that if you deliver phages to at least cells in the laboratory and you look at, these are human cells, so these are not bacteria, you can find entry. You can find that phages enter human cells at a very low rate, and it could be pH dependent for some phages. So if the pH is a certain value, it'll happen more or less. So that might actually be useful if you consider something like the CF lung, which is a different pH than the normal human lung. But the point is, This is kind of good news, bad news. So if I'm delivering a phage and I'm trying to kill bacteria, but your own cells are occasionally sucking it up, then it's going to lower my dose, okay? So that's kind of the bad news. I'm delivering less of what I expect to work. The good news would be, in these experiments, the cells mount an interferon response. So the phages don't seem to be able to replicate, but it will cause human cells to make more interferon. And if that is just generally happening in phage therapy, then that might be kind of good news because you are pushing the immune system to help, okay? So it becomes very realistic for people like Joshua to build that in to the models. The antibiotic resistance problem is a One Health issue. You might have heard this because it's happening in non-humans. So there's lots of increasing evidence that antimicrobial resistance is happening in plant and animal agriculture and aquaculture. So necessarily, at some point, when it gets bad enough, it will threaten food security. I think that that's a no-brainer thing to say, and I stand by that statement. We live with many domesticated animals in our homes. I mean, you may live on a farm, some of you, I understand that, but maybe more of you are likely to have cats and dogs in your homes, and increasingly they have antibiotic-resistant infections that are hard to treat. And I have some interest in conservation biology. I'm sort of into species and species preservation, and I was kind of shocked when I started hearing from veterinarians about large animals in aquaria and zoos that have these problems, and they can't be replaced if they die, okay? Why? Because there's a lot more rules now about taking animals from the wild. If you have a beautiful sea turtle die in your aquarium in Miami, that's game over. You're not going to go get another sea turtle necessarily. So we worked with veterinarians on that problem, and it worked well. Otherwise I wouldn't be talking about it. So this is something that is promising as a technology that can help us for food security and in conservation biology. Last would be access and sustainability. So a lot of what I told you is how phage therapy is moving forward in places like the United States and Canada through personalized medicine. So people can get access to life-saving treatment or treatment that could save them from undergoing pulmonary failure and then get a transplant. But, who knows about it? I mean, now you know, but I'm not expecting all patients and physicians are aware of potentially life-saving technology. That's just a statement about biomedicine. I think that's a statement I'll stand behind. And not all patients happen to live close to New Haven, Connecticut, or Pittsburgh, or San Diego. And there's like about 5 places in the United States where you can really expect to have this in your backyard. And if you can't travel to those places, you won't get access to it. So this is automatically a bioethical issue. And we're starting to write papers on it. One of my former postdocs, who's now at University of South Carolina, Jordan Lewis, he's an evolutionary biologist and also a bioethicist. Turns out when he was at Emory, he took some classes from Gerard Vong. So Gerard is a co-author on some of our work on the bioethics of access. And last would be, there's funding challenges in this. I don't know what percentage of what I showed you came kind of out of pocket, but you can't get a grant that I know of to really do compassionate therapy. You can't do personalized medicine necessarily with a grant from a federal agency in the United States, for sure. So that means the compassionate treatment that we do is not really funded that much by sponsored projects or external awards, and we have to kind of carve out our own resources to do it, and that's not endless, right? That has a finite cap to it. The clinical trials that prove whether or not something can go to a drug labeling phase are egregiously expensive. And some of that we found in our first clinical trial is that we had to pay another company to make the phage doses. And it was so expensive. So now we have a phage manufacturing facility. It's not very large, but we decided to build it because otherwise we couldn't affordably do it. And I do worry about the small consumer being priced out. So basically a lot of this will not go forward because you hit a clinical trial and it's just too expensive to happen. So my prediction is that support can and probably will come from philanthropy in the near future, because this is already happening, and it seems to be pretty crucial, especially when you're sort of out at the edges of it. If you've got a bacterium that you suspect is interacting with the human nervous system, maybe Parkinson's disease and other neurological problems seem to have some, at least hints of evidence that the bacteria you carry in your microbiome are out of whack some way. Either there's an invader or there's what's called dysbiosis, And yeah, you could take a phage and bring them back into alignment. That may or may not work, but you may be able to take in principle the same approach I've been telling you about trying to stave off some of those problems. So that is very ambitious and early stages, but it relies on some resources to have it happen. Last thing I'll say is Ben Chan in my group, he was featured in this very nice documentary for which I'm not in it, and I don't need to be, so don't expect to see me, but it's called The Good Virus, and it's really about how in low and middle income countries, this technology can be very good because it's not that expensive. And we have to remember that as you're grooming this technology, not only do we have access issues in our own Western, pretty wealthy countries, but there's entire countries that would love to have this technology for things that are problems in their backyard that we frankly don't care about, because they're not problems in Canada and the USA. So that's something else to keep in mind. If you can dial up somehow the good virus, just contact me. I'll send you a link to it if you want. All right, so thank you to the people who did the work, and our generous funders, and my lab group through the years, and thank you for your attention. Yeah, you go.
Thank you, Professor Turner, for a wonderful talk.
Sure, thank you.
So, hi everyone, I'm Allison Hill. I'm a faculty member also in the Department of Ecology and Evolutionary Biology here, and I'll be kind of taking us through the next section of our event tonight, where we'll have some discussion on the topics that Professor Turner has brought up. So first, we have a few minutes for audience questions. If you'd like to ask something, please come up to the mic, and in the meantime, I'll get our panelists to come up and sit down, so you'll be ready for the panel discussion. But if anyone wants to come up, ask a question to Professor Turner, feel free to come to either of the mics here.
Hi.
Hi, thank you. Thank you for the amazing presentation.
You're quite welcome.
In terms of human immune response, how long would it take the human immune system to attack the phages?
Yeah, great question. So the data are not general yet. We don't know. So for some reasons, you can deploy some phages and the human immune system never catches on. So we've had, I'll give you anecdotal evidence, there's some people we've treated and the therapy went very well, but maybe 3-4 months later, the physician contacts us, oh, she's not doing as well, we see the bacteria creeping up again in number, can we deploy that phage again? Like, well, all right, we're approved. We can do it. It's probably never going to work because you've got an immune response, and it worked. So at least in that phage, in that person, there was no neutralizing antibody formation. And yet in other examples I could give you, there was almost an immediate neutralizing antibody. So we have to understand that better, but it's maybe this evidence of phage to phage that could be quite different. Yeah, it's a good question. Yes, sir.
Go ahead.
Resistance is the cure. The pyramid of memory. What is the most useful biological formula of relevance that makes a useful difference in and around the workplace? The liturgy of the ecosystem, syntax, synapse, the evolution of the syllogism, the buzzwords of the advertising of the evolution and revolution of biology. The atom is 1,867 times bigger than its electron. Blink, your DNA is having its picture taken by its RNA. In between the mind's camera, the DNA and RNA is an atom of information that might have a handle on the revelation of how to stop the next market crash, or man's next biological discovery that will cure the next epidemic. Or it might be the handle of the practical axiom of this brilliant biological lecture. What was your greatest discovery on your winding, meandering trail of biological innovation?
Thank you. So I am constantly surprised by what biology shows me. And I think it's very difficult to make predictions, but it's important to make hypotheses, have them supported or refuted, and learn from it. So I'm happy that I've spent a lot of my career being wrong. I learn more almost from being wrong and having my hypotheses refuted for me to go back in and reconfigure my ideas and my understanding of the system and to probe further. So I would say I'm constantly surprised by what I see.
Great.
Thank you. Hi.
Hi, yeah, great talk. I was really struck by the comparison from your clinical data on the amount of phage resistance in the pre and the post samples. And why aren't more of those bacteria already phage resistant? Don't they already find phages in their environment that they have to cope with? Is it that's not the right phages and they just haven't seen these ones?
Correct.
What's going on there?
It's a big biodiverse phage world, including the phages in your body as part of your microbiome. So when we have this invading bacterium and we find a phage in the laboratory that can kill it, I have to prove that they're susceptible, and then if I probe further, I should see the evolution of resistance of what I'm saying is correct. But that has nothing necessarily to do with other phages that they encounter and their susceptibility and resistance to those. So it's many different outcomes are there. You could have generalized resistance, generalized susceptibility, and we're learning more and more about it all the time. Good question.
Just in a matter of time, we're going to move on to the panel now, but we'll have some time right after this for more questions. Don't worry. Great. Okay, so I'm going to introduce our panelists that we have here today. So Dr. Greg German is a professor of bacteriophage therapy and innovation here at the University of Toronto. He's also a medical microbiologist at Unity Health, and he's the co-founder and clinical director of Phage Canada, which is an organization that promotes the use of phage therapy throughout Canada and also around the world. We also have Professor Arjuman Siddiqui here. She's at the Dalai Lama School of Public Health, and she's also the Edwin Long Professor of Child Policy Research. She is an expert in health inequities and understanding how social and health policies can either help solve or perpetuate disparities in health. And then we have Professor Nicole Medeo from the Department of Ecology and Evolutionary Biology here as well. She's an expert in pathogen evolution. She studies malaria and understanding how pathogens evolve drug resistance and virulence and other important traits. And also as a professor here, she teaches evolutionary medicine to hundreds of undergraduates every year. So thank you all for being here. We're going to start off with some questions for the panelists, and then we'll open it up to more discussion. So maybe we'll start off with Dr. German. Can you just tell us a bit about what is the status of phage therapy in the clinic today, in Canada or more broadly, and where do you and Phage Canada hope to see this in 5 years from now?
Thank you so much for the question. And the technical difficulties are now corrected. So one of the first things, I tend to be an interactive type speaker, and I did want to take a quick second to answer this question. How many cases of phage therapy to treat urinary tract infections have there been since as far as we know? And raise your hand if you think it's 100 cases. 200, 500, 1,000, 1,400, 1,450. So whoever raises their hand for 1,450 is correct. And so this is going back all the way to 80 plus years, including work that we've done and will be highlighting further. And the safety profile, the eradication capabilities, very strong. Single-use cases. Our biggest challenge right now is, are we doing single-use cases? Are we doing precision therapy? Are we making it a medication like aspirin where we have to churn it out in good manufacturing practices? And are we doing randomized controlled trials? So as we see in our states, in the United States, there's a lot of single-use cases, upwards of 1,300 of them. Some people will even calculate 2,000 of them, but they're not stitched together in a systematic way. Dr. Pierney's paper was talked about in Belgium, where they had 100 cases picked in a row as part of a study to make sure there wasn't something called publication bias. And they also showed 80% effectiveness. Now, that's also using a personalized approach, where we're kind of talking about, can we make it so that we have the best key, lock and key perspective, rather than a gigantic master key that doesn't work all the time or can quickly develop resistance. So we're still working through that. There have been 3 or 4, maybe 5 randomized controlled trials in small groups. One a while ago for Pseudomonas in the ear that worked out fairly well. Another called Phage burn that didn't work out at all, in part because they didn't actually give any phages because of how they calculated things. Duh, it didn't work. And then urinary tract infections, we're seeing some signal from a company out of North Carolina, Locus Biosciences, where in a small subgroup, part of their randomized control trial, still small numbers, they also showed 80% effect with a very broad cocktail. I can offer that for myself as the international lead for the out of Unity Health Toronto, and with 9 other clinical sites, we are greenlit for a Horizon project for 15.6 million euros over the next 4 years. So we're also, in working with other colleagues, greenlit for another for prosthetic joint infections. Now we're talking about 220 to treat female urinary tract infections with E. coli, or for this larger project, 450 prosthetic joint infections with Staph aureus as a gram-positive one, and frequent cause of prosthetic joint infections. So all that being said, there's tremendous work that needs to be done, and I'm happy to answer any more questions. And then also, please check out Phage Canada, because here's your chance to become involved. Even the public, we have 10 public members of our 110 scientists and clinicians that are part of it, and so there's a great opportunity. Thank you.
Cool.
Wonderful. Thanks for that update. Professor Siddiqui, so we know that antibiotic resistance, like so many infectious diseases and other health concerns, disproportionately affects marginalized groups. So, both in Canada and the US as well as all over the world. So even at this early stage, what should we be thinking about? What should we be doing to help ensure that the rollout of phage therapy really helps sort of improve health equity issues as opposed to kind of exacerbating them?
Great, thanks for that question. I don't know if I can follow giving a gift to the audience, but you don't play fair. I think we learned something from the COVID-19 pandemic, which was that when we unfolded a vaccine strategy, we did so without considering the groups that needed it most, which were primarily racialized people, primarily low-income people. And we unfolded the vaccine strategy in a way that located the main places to get your vaccine in high-income white areas. And so I think what we learned from there is that we need to consider equity right from the start. We can't take it lightly. We can't imagine or assume that these therapies or anything else will just somehow magically osmose their way through populations. It really has to be an intentional and deliberate strategy, and one might argue that it should be foundational, that without it, we sort of have failed if we don't find a way to make these equitably accessible.
Great, thank you. Professor Medeo, when we think about evolution in the context of infectious diseases, we tend to think about it as a bad thing. So whether it's antibiotic resistance or new flu strains or new COVID variants, but how are we also harnessing evolutionary thinking to try to find solutions? So is phage therapy just one example of this way that we could reframe evolution in medicine?
Yeah, that's a great question. And you kind of highlight a point here that these are evolutionary problems. Like these are issues that are outcomes of evolutionary processes, and so probably evolutionary biologists will have something to say about how to deal with them. And Professor Turner said in his talk that pathogens are going to evolve. Like that is going to happen. That is not an open question. So the question is like, how can we deal with the evolution? Can we steer their evolution in ways that benefit us? And so he presented like one really nice way of like exploiting these trade-offs. But a lot of folks are thinking about a bunch of different possible ways of dealing with this selection problem. How do we slow the selection for resistance or shift selection? And so there are other examples. There's 3 that I want to tell you about. One is rather than trying to kill the pathogen, can we give drugs that deal with the damage that pathogens cause? So improve the tolerance of the host to the infection. And by not actually killing the pathogen, you're not making this a life or death thing, evolving resistance. So that reduces selection for resistance to these drugs. That's one option. Another option, which I think is really cool, is thinking about different ways of using drugs in combination. And Professor Turner mentioned synergistic combinations of drugs and phage. But when it comes to drug combinations, there's a bunch of people who think that actually antagonistic drug combinations are really useful in the face of resistance evolution. So these are, you put two drugs together and they work worse together than they would on their own, which means that if you use the two drugs together and a pathogen evolves resistance to one, that amplifies the effect of the other drug that's left. I think that's a really cool idea, so that should slow resistance evolution too. And then the third idea kind of deals with maybe another trade-off, sort of like ecological trade-offs. Professor Turner mentioned resistance is going to be rare often in an infection, and so folks are thinking, can we change the dose of drugs or the timing of drugs so that we kill pathogens more slowly? Most of the pathogens are going to be drug sensitive. If we kill them more slowly, they can outcompete any resistant pathogens that happen to be in an infection. And that idea doesn't just apply to infectious diseases, that also applies to cancer. In the cancer world, they call it adaptive therapy. Rather than trying to cure cancer, cure a tumor, can we just reduce its size, sort of maintain its size, give lower doses of chemotherapy or spread those doses out so that you're kind of like maintaining a pool of sensitive cells that suppress the growth of any resistant lineage. So I could go on at length about this. As you said, I teach a class on this and give many lectures on these topics. But there's some examples of other ways of kind of harnessing evolutionary thinking, thinking about selection, and adjusting treatment based on those thoughts.
Right. So maybe just to build on that before getting back to some of these other really important topics that have come up. So, some of these super tough cases that clinicians have to manage, these really complicated infections and figuring out treatments. I mean, these are evolutionary problems, and yet, pretty much nowhere in medical school, in pharmacy school, do people learn about evolutionary ideas. I mean, even here at U of T, we have our department. It's on the complete opposite side of campus from where everything medical and public health is happening. Like, is this a problem? Do we need to change how we approach kind of education across these different realms of biology to, get at some of these, really difficult medical challenges? And that's sort of a question for anyone in the panel who has thoughts on that.
Sure, I'll speak up. So I was delighted when the MCATs decided to put evolution questions on there. It enforced that exactly what you're saying is true. We need to have a broad perspective and understanding of biology to do biomedicine, because evolution is always going to creep in. So I and others were contacted about generating questions. I never quite looked to see whether my questions made it on the exam, but the point is I was just happy to help them go forward with that. Nevertheless, what you're saying is very true. It's not easy to become a physician and make it through medical school. You have a packed schedule of classes and activities, and to put something else in, it's always been seen, no, there's enough, there's enough. So this has been a very tough road. I think that there are some incremental advances, like this MCAT example I gave you, but it's been tough, I have to admit. There's a society called the International Society for Evolution Medicine and Public Health, and I was president of the society, and it's been very vibrantly that public health and evolution experts are active in that society, but physicians they're busy seeing patients, and they have plenty of conferences already to attend. So it's been a little hard even on the intellectual front to try and synergize across, but I think we just have to keep trying, because it's a big kind of complex biological world out there, and you want to have all the tools possible to grapple with the problems, in my opinion.
Antimicrobial resistance is a crisis of economic sustainability. When it takes $2 billion in 10 years to produce an antibiotic and bring it to market, no one wants to function or work through that. We need to use phages as an alternative method. When I go on TEM talks, which is Temerity Faculty of Medicine with Andre Picard, and I talk about how we need to talk about this at the dinner table. When I write in Maclean's that we need to use every tool we can, we actually have to get the whole world together, not just one thing, and it has to be with the world of AI and ethics and biotechnology with microfluidics and designing the machine that's going to actually make those phages even smarter, safer, and more effective. And there's that proposal by 2030 that machine will exist where you just put in the isolate, you took the swab of the ooze and you put it in, and then bing, 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 out comes out the phage product. So we can get to there. We actually have the tools. It's now miniaturizing it, and it's working together to use it wisely and sustainably.
Go ahead. Just very briefly, I think there's a sort of fundamental problem in terms of making sort of modern day education in medicine and in public health, for that matter, all too technical and less sort of about fundamental science. And that I think we need to be a little bit more open to the notion of fundamental science and curiosity, even in fields that we think are sort of deeply technical and require, as Professor Turner was saying, a lot of technical training. We still should make some room somehow for some of this more curiosity, innovation-driven work.
Sure. I think, people have been talking about this for literally decades. How do we get more evolution in a medical curriculum? And what would we take out if we put more in? And, maybe I totally agree with you, Arjun, but maybe we're not moving the needle on that. But there's two other options here. One is do it at a different level. As you point out, I teach this evolutionary medicine class. I know a couple of my students are in the audience, and every year I know some of them are going on to med school. And several of them have said to me, I'm going to take my evolutionary thinking with me. And I'm like, thank you. I don't know what you're going to do with that, but I hope you do, hope you're a great doctor. So there's that. We can get them at a lower level. Evolutionary biology is interesting, and so we can teach it at the undergrad level. And then the second thing is collaborations. The work that I was talking about that I mentioned, the other approaches for dealing with antibiotic resistance, that's evolutionary thinking, evolutionary biologists working with clinicians in several of those cases, and we can always do that. I think that is a big opportunity, that growing the links, as you pointed out, the buildings are on different parts of the campus, but growing those links, I think, is another way to do this.
Cool. Great. So maybe going back to the very beginning, you got to this at the beginning of your talk, that the idea of phage therapy has been around since the 1930s, when, really the dawn of microbiology and molecular biology, but, it didn't take off in most of the world. And, my understanding is there's a combination of reasons for that. We need to be able to diagnose things precisely and quickly. We need to be able to manufacture phages, and we need the public to be cool with the idea of taking phages. So what is different now? Have we solved those problems? How are we going to get this to be widespread now compared to sort of the last attempt at this almost 100 years ago?
I'll start first. So do you know that you look at your urinary tract infection report and it says a bacteria and it says then the antibiotic, and that's SIR, sensitive, intermediate, resistant. We don't have that mapped out for phages, even after all this time. And I sit on an international group of people who are coming up with interpretive guidelines. And we don't have the randomized controlled trials. All of that other basis for that urinary tract infection for SIR has been based on decades of work. So we really need to figure out what it really means to be sensitive, intermediate, resistant, and that'll be one of the major steps. And we're only about 1 or 2 years away. You asked about what's going to be in 5 years. That's one of them. Registries are also going to be helpful, where people can self-enroll in potential processes or send strains for susceptibility using standardized methods. And I'll give you back my time.
Thank you.
Yeah, I think building vast libraries of phages that are actually shared with others is important. Right now, there's still a lot of expectation that people would make some money on it, and I don't think we need to approach it that way. We need to make sure that we are planning for the future. Humans are very, we respond well to things. We're not very proactive about things. And you said earlier, your comment about well, the antimicrobial resistance problem is indeed a problem, and I hope you appreciate that from either your prior knowledge or from my lecture and what you're hearing. But you don't really hear about it on the news, okay? And so if I just stop somebody on the street and I say, is cancer a problem? Of course they're going to say yes. Why? Because they've had it, maybe, or their relatives have had it, or their neighbor had it, or their dog had it. And they just know it's very dangerous. But I don't think people appreciate the antibiotic resistance crisis yet, because not everybody's living there yet. Some people are already. So I literally have heard people in the media tell me, oh, you scientists, you're not giving the right message to people. And I don't think we deserve that blame. I'd rather push it on them. Well, the media needs to figure out how to convey that this is an issue, and it's going to grow, and it's going to hit the average household. So that was a bit of a rant, and I'm not sure why I went on it. But the point of part of the issue for the growth of this technology is the inherent kind of misunderstanding about it. It is a biological drug, and we know antibiotics. We know how to produce them and then measure what you produce, and it is within bounds. I mean, it is a chemical you're producing. You should be able to figure out what you're producing. There's an inherent sloppiness to phages because you're always going to get the genetic variation around what you produce. And we have to embrace that. We can't be fearful of that. We have to understand variation. So I'm very intrigued about what you said about coming up with the rules and the rationale behind these therapies and how this kind of genetic uncertainty or the cloud of mutants that you would expect in your doses shouldn't matter, and they haven't mattered in the personalized medicine that we've done, but they're there and they're not going to go away. So I don't know how the kind of same rules that we've created in antibiotics and other drugs per se would actually work for biological drugs, full stop. We have to come up with our own kind of chill ability to understand what a biological drug can mean.
If I could just add
please.
on to that a little bit. So how many drugs do we use to treat HIV patients with at one time? How many drugs do we use to treat tuberculosis patients or even drug-resistant tuberculosis patients? It's like 5 or 6 drugs. And what the intention is that you knock down through an evolutionary process and use different targets in order to get to a certain point. There's also definitions of the biological load. Certain infections have a higher biological load of bacteria, or they have more quiet, hidey places like biofilms. So we may have to engineer multiple phages and multiple strategies. Maybe it is 3 using different targets to laser at different areas. We know that phages are generally safe, but it's that potential target, and then maybe you have to carry on what the predictability is of the mutants and have targets built for them. So it may be that phages isn't just a 1 and done or a 3 and done, but it's an ongoing process of personalized medicine, and that's pretty exciting, but also scary for as we work through the costs and the AI and technical challenges that encompass this.
No, but I did want to hear from Arjumand, actually. Like, just in terms of the end of your question about, like, how are people going to be cool with taking a virus as a treatment?
Yeah, I don't know that I have sort of a well-founded answer. My general thought about all of this is that I think we do need to take education really seriously from the get-go. And I'm reminded of an economist and sociologist duo I know who did a trial to determine whether sort of gifted education in very young children who were very disadvantaged and would have otherwise been strained remedial, how well they were able to take up the most gifted curriculum, and they did splendidly well. And so I just think we need to be a little bit more sort of engaged with making people aware very early on.
I would say we're also dealing with challenges with sagas, that people have been in infection for 2 years, 3 years. And if we know anything from fecal microbiota transplant, which is otherwise known as something else, they're fine with it. And that's 10 times worse than phages. And what's interesting is that they're actually getting a big dose of phages when they get that transplant as well. So I think people will recognize the inherent safety in it. And yeah, that 1,450, there were 8 with side effects, incredibly low. And even in a subgroup of just phages alone, not phages antibiotics, it's only like 1 or 2 of the 500 that had side effects. So I do think it's far better than antibiotics is what I would try and say. And just getting back to the last point of how are we supposed to sell this? And I was at a big meeting in Luxembourg for TATFAR, and it has like CDC and European Union, and it's like, stop killer bacteria. We just need a really strong statement.
Yeah.
and
Yes.
equal phages. So that's what, and that may be too brash, but we do have killer bacteria, and it doesn't have to be drug resistant. We're actually seeing ones with just biofilm, prosthetic joint. I want to keep my leg. Well, phages may help you keep your leg without having to go through 7 revisions. So I want us to go back to sagas. We need less sagas.
Great, thank you. I'd like to open it up. If anyone from the audience wants to ask any questions, for the panel as a whole to answer, want to come down? You have something? Yeah, just, sorry, we can't, I don't think these mics are movable.
That's okay. You can move.
Thanks for the lecture, even though I understood like 20%. I was just curious, where do AI tools and large language models actually add value in optimizing phage therapy, given complexity of bacterial evolution?
Yeah, so one way would be we're getting, I mean, it's easier and faster to get genomic data. I think most people know that. So if I show you that long list of a library of phages and some clinical strains of bacteria, we know the genetics. We know the genomics explicitly. So in kind of a challenge like that, which phage can kill which bacterium, the question is, well, what's driving that? Is it purely genetics? And if it is, then you should be able to take machine learning and AI tools to it to say, here's a genome of a bacterium. Can I know from that genome alone which phages should be able to kill it? And when we did that many years ago, and people like Adam Arkin, he does a lot of that work, and it's actually pretty predictive, 70, 80%. But the problem is, what's up with the missing 20%? And so one thing that I'll leave you with, I'm not going to go on at length about this, is that bacteria often have other things in their genome aside from their genome. And that could be phages that are hitching a ride with them that don't kill them, and they're inherited by all the offspring that the bacteria makes. They can be responsible for closing the door for a phage to come in. So the sheer genetics of the bacteria alone won't necessarily help you. It could be what the bacteria are carrying around with them as plasmids or prophages or other mobile genetic elements that help us understand the missing 20%. And that's where it gets really hard, because if you all were bacteria and you're in the same population, I wouldn't expect all of you to have these mobile genetic elements that may dictate your susceptibility or resistance to some lytic phage. So that's quite a challenge, but as we continue to use machine learning and other tools, we might be able to encompass that and make better predictions. I hope that's helpful.
Can I just love the prophages? It gives you like a map on how to enter the cell. Another AI angle is protein folding, and we'll know more and more through the years. We're both tool C lovers, by the way. Like, I identified a phage during my PhD dissertation before medical school. You know, we have U136B founder here, the other tool C phage. And all that being equal, knowing how to target that specific thing with making the gene to make the amino acids, to make the protein and folding a certain way, those are AI tools, AI engines.
That's great.
Go ahead.
Thanks for the references in terms of the CF and lung cases. My question is there any promising phage therapies or research in terms of another lung disease called MAC? So,
what is MAC? Oh, yes. So I'm not an expert in that, but essentially the vulnerable patient groups, of which I mentioned CF, but there are far more people who have, for example, chronic obstructive pulmonary disease, COPD, and they can be equally or more vulnerable to some bacterial infections coming in, and these are of a variety of sorts. So I'd say that if you look at the laboratories that are studying what I do, you know, they tend to be specialized in approaching some particular infection problems, and that's a long-winded answer to say yes. There's a lot of activity going forward in just about any, he says cautiously, I'm not quite sure if that's a true statement, but almost every major respiratory bacterial pathogen. The main issue is, can you find phages that either do what I do, or can you find them at all? So a dirty little secret of phage discovery is you might have a bacterium that you want to find phages for it, but it's not necessarily you would find them if you check every waterway and soil sample you've got. And that's a bit of a mystery. phages are incredibly diverse, but we have to be careful about the way we discover them is by growing some bacterium in the laboratory. And so first of all, you can't grow all bacteria in the laboratory. So you have to think of other ways to capture that phage diversity. I'll leave you with one. It's a little technical, but it's easier and easier to go out and sample microbes and just come up with a profile of their genetics and genomics. It's called metagenomics. And increasingly, you could see something that's like, oh, that's a phage, but I can't grow it. So there's a way to print the DNA and make the phage anyway. Okay? So there is some hope for hard targets.
Can I try it as
There
well?
is.
Graham Hatfill, in a major article, looked through 22 cases of Mycobacterium disease. And we talked about philanthropy. He's a Howard Hughes scholar and gives away the treatment for free across the world. And so he's probably the only person that's in this space providing this therapy, including one of our Canadian cases.
Yes.
So tremendous potential, and it just goes to show how we need to work together to make that work for one disease that can be done for others. Unfortunately, he has a great deal of challenges that there's not always a match. The last thing I want to say on Mycobacterium, there's also interest in tuberculosis in Africa in particular as another strategy, but it's still in the early phases.
Go ahead.
Oh, okay, so something that you mentioned earlier that I really, like to hear was that, as we're developing these treatment strategies, we want to make sure that we're having, equitable access from the start. Something I was wondering to, ask, you and other members of the panel was, what are some ideas you folks have on how we can go about ensuring that this doesn't exacerbate healthcare inequity?
Yeah, I think there are a lot of structures in place to try to mitigate inequities. Working with communities, there's kind of no substitute for, and involving communities very early and very often in the education around this, in the process of how these things are manufactured, utilized, et cetera. All of it sort of from day one, having input is a big deal. On the other hand, the inequities that could occur for technologies like this aggregate up to inequities in just about everything related to health and healthcare, as you probably well know. And so some of the solutions are also very macro, are very sort of designed to think about what fundamentally causes inequities. So there's things we can do to mitigate specific inequities. There's things we can do that would sort of do some sort of more meta mitigation. And then there's addressing the fundamental causes themselves, which is, again, very sort of structural.
Well said. Yeah. I think structural issues are going to be a big impediment, but I think...
just to add a couple extra things, like food security is going to be a big issue. Antimicrobial overuse is a challenge, and phages can get around that potential. From the clinical trial side of things, it's having patient advisors sitting at the table every single step of the way. I'm focusing on urinary tract infections and women between ages of 20 and 80. And that's an area that has been unfortunately stigmatized when it comes to urinary tract infections, and we hope to address that. I do think it's getting together again.
One thing I'll add about urinary tract infections that I found very dismaying is that somebody contacted us and she'd had a 40-year running urinary tract infection, and I'll throw the US FDA under the bus. They didn't call that an emergency. So that's a quality of life issue. So another quick example would be a lot of people from a very young age are administered antibiotics for life because it's used prophylactically, and it really messes up your gastrointestinal system and your microbiome through off-target problems. So I would say minimizing antibiotics dispensed to children. The pediatricians, when I tell them about my work, it's like, if you can do that alone, you're improving a lot of quality of life for people and a better horizon for them. So there's a lot of, you have to think creatively about where the technology could be useful and implemented. So thank you for that.
Thank you very much.
All right, so it's time for the third stage of our event for the evening. So there's a reception in the lobby. You are all invited. You can exit this way or back here, and feel free to come and ask any questions that you didn't get a chance to of any of the members of our panel or our speaker here today. So I just wanted to thank everyone again. Thank you to the Martin family for supporting this. Thank you, Dr. Turner, and thank you to our panelists for a great discussion.